Ever wondered how DNA replicates? Well, it's a fascinating and intricate process that happens in every living cell. DNA, the blueprint of life, carries crucial genetic information. It's like a coded manual that instructs cells on how to function and grow. Now, just imagine, this manual has to be duplicated flawlessly billions of times over in every single cell. Quite a task, right? Yet, nature pulls it off with remarkable precision. So how does this process actually work? Stay tuned to find out. So, DNA replication begins with the unwinding of the DNA double helix. This is where the magic happens. Picture this: two single strands of DNA, each serving as a blueprint for the creation of a new strand. This is made possible by the enzyme DNA polymerase, a master builder of sorts, which synthesizes new nucleotides to match the template. These new nucleotides are added to the free 3' hydroxyl group provided by the primer in each single-strand DNA. Now let's talk about the leading and lagging strands. The leading strand runs from 3' to 5', and DNA polymerase adds nucleotides from 5' to 3' direction. As the replication fork moves forward, the addition of nucleotides is a continuous, smooth process, needing only one primer. But the lagging strand is a bit more complicated. It runs in the opposite direction, from 5' to 3'. Here, DNA polymerase can't add complementary nucleotides to the 5' end as the replication fork progresses. This means multiple primers are needed, leading to nucleotide synthesis in fragments. These fragments are known as Okazaki fragments. So, to sum up, the leading strand uses just one primer and synthesizes nucleotides continuously. The lagging strand, on the other hand, uses multiple primers and synthesizes nucleotides in a discontinuous fashion. In this way, two new strands of DNA are synthesized, each complementary to one of the original strands. Now that we understand how DNA replicates, let's recap the key points. DNA replication is a crucial process that begins with the unzipping of the DNA double helix. The single-stranded DNAs then serve as a template for the creation of new DNA molecules. DNA polymerase, a key enzyme in this process, synthesizes new nucleotides to match the template. It's important to note that the addition of these nucleotides occurs in a specific direction, from 5' to 3'. The leading strand requires only one primer and is synthesized continuously. However, the lagging strand, due to its antiparallel orientation, requires multiple primers and is synthesized in fragments, known as Okazaki fragments. This intricate process ensures the accurate replication of our genetic information, preserving it for future generations. And that, in a nutshell, is how DNA replicates. A complex process, but one that is essential for life as we know it.